You're at the cutting edge of cardiovascular sciences. I'm John Cook, professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist. And today we have a very special guest. I'm joined in the studio by Dr. Anahita Mojuri, who's going to introduce our guest today.
Yes. Hello, everyone. Today we have Dr. Nicholas Lipper with us on the show. Dr. Lipper is a professor of vascular surgery and cardiologist, chief of vascular medicine, and director of vascular research at Stanford University. He is a practicing physician scientist interested in the intersection between cancer and cardiovascular disease. His lab pursues their conditions with a combination of translational approaches from the field of human genetic, bioinformatics, molecular biology, and marine cardio-oncology models. Their goal is to advance finding from bench to bedside to address the world's two leading killers. He also directs the Stanford Vascular Medicine Translational Research Program, which works towards targeting these processes in humans, and co-direct Stanford's T32 on vascular diseases, which aim to train the next generation of cardiovascular researchers. The title of his talk today is Immuno-Cardiology, Bringing Pro-Efferocytic Therapies from Bench to Bedside.
Nick, it's great to have you on the show. Great to see you again. You've done some amazing work at Stanford, and it's quite incredible because you're also a practicing clinician. Today you're going to be talking to us about the efferocytosis story and its role in atherosclerosis, but tell us a little bit about your practice of medicine. What kind of patients are you seeing now, and tell us a little bit about your patient care.
Sure. First, let me thank you so much for inviting me on. This is really an honor. John, as you recall, I was one of your first trainees on the Vascular K-12 program many years ago at Stanford. And under your tutelage, I learned how to not just take care of cardiology patients, but also to love blood vessels outside of the heart. And so, in your legacy and that of Victor Zhao's and those who came before us in the vascular medicine arena here at Stanford, I care for patients with peripheral arterial disease, aortic aneurysm disease, carotid disease, as well as other vascular disorders affecting the lymphatics or the venous system. And so we have a really wonderful practice here trying to provide, quaternary care for both bread and butter atherosclerotic diseases, but also those that are less common, and really trying to help these vascular patients preserve life and limb.
That's spectacular. We certainly need vascular medicine. Vascular disease is a major cause of morbidity and mortality in the country, and we need more people like you, Nick. I'm so happy to have handed off the banner to you, and you've carried it a lot further than I ever did, so thank you for that. We're now going to hear a little bit about your really stunning work that is interesting from a basic science standpoint. It's fundamental insights into understanding atherosclerosis, but also some translational application that's going to be useful to our patients in later years. So, Nick, please begin with your talk, Immuno-Cardiology, Bringing Pro-Efferocytotic Therapies from Bench to Bedside.
Well, thank you again, both. I really am quite honored to get to be here and to share this story, which is really a pretty exciting, bench to bedside translational journey that we're not quite finished with yet, but we hope will bring new medicines to patients with atherosclerotic disease. Just by way of background and disclosure, I'm the co-founder of two companies, and I will be discussing some both published and unpublished work that's emanated from these groups. So, John, as you mentioned, as vascular medicine specialists, we think a lot about atherosclerosis, which of course is the disease underlying heart attack, stroke, and peripheral arterial disease. One of the things that struck me early in my career is that, while we've gotten really good at addressing the risk factors for atherosclerosis, and we've had, incredible advances in antiplatelet and anticoagulant agents that will prevent clots from forming on top of the plaque, really most of what we do is either mechanical in nature, opening up blockages with stents or bypasses, But you could make an argument that we currently have no therapies that actually directly address the pathobiology in the vessel wall, and certainly nothing that meaningfully reverses the buildup of plaque, the way that oncologists might think about how to get a tumor to shrink or go away. You might argue that this doesn't matter because we've had decade after decade of improvement in cardiovascular medicine. But I would argue that we're reaching the point of diminishing returns. And after the major advances with smoking cessation and lipid lowering, we've seen a plateau in many of these curves. And in fact, there's been an uptick in some cardiovascular diseases related to the obesity epidemic and heart failure. And so we really have a lot of work left to do. If you think about what the pharmaceutical industry has been focusing on, they've really focused on addressing the established risk factors over the last several decades. And they've shown pretty successfully that if you continue to drive these conditions lower and lower, like blood pressure or LDL cholesterol, you can have some incremental benefits. But I would also argue that we are really getting to the point where we're getting less and less juice to squeeze out of the fruit. And the absolute risk reduction that we're observing is fairly modest. And I think that that's the reason that we see this, really striking residual risk that's not addressed with our current armamentarium. Now, this is not to be pessimistic, and there have been some really important advances recently, particularly with new targets that will lower remnant cholesterol and triglycerides with the advent of ApoC3 and ANGPTL3 inhibitors. We're seeing really exciting data with new genetically validated targets like Lp(a). And so there is much coming, but you could argue that most of what we've done has been related to lipid lowering. So this begs the question of what else is out there. And, as you're well aware, many people have been thinking about the inflammatory component of cardiovascular disease, which may be independent of the lipid risk. And so through really pioneering work from Paul Ridker and Peter Libby and others, we started to think about how we need to address these two different types of risk in parallel. And you get a sense that this really matters because whether or not we get our patients to their LDL targets is of course important, but if they remain inflamed, their risk is high. And so there's been a lot of interest in whether or not anti-inflammatory therapies will be helpful. And I think that this audience is probably well aware of the landmark CANTOS study, where they used an antibody which blocked interleukin-1. And they showed that this could very potently quench inflammation as measured with CRP. They could do this without having any influence on LDL cholesterol, so showing that this is truly orthogonal to our current established risk factors. And, the takeaway was that this did reduce adverse cardiovascular events as predicted. But it also had an Achilles heel, and they found that when you did suppress the inflammation and you had the cardiac benefit, you were also having immunosuppression, and there was a significant increase in fatal infections. And this is one of the reasons why this drug was not approved for cardiovascular prevention or secondary reduction of endpoints. And so, we are still working very hard on this concept of anti-inflammatory therapies. There are a number of new approaches that are being tested, including data which suggest that colchicine may have an anti-inflammatory effect that's beneficial. There's a lot of interest around interleukin-6 in particular, following two provocative phase 2 trials, and these have now advanced to phase 3. So we eagerly await novel anti-inflammatory therapies. But it remains to be seen whether or not they will also have immunosuppression and have some of the Achilles heels that prevented canakinumab from being advanced to the mainstream for secondary prevention. So this begs the question of what else is out there and what else we can do. And as you know, John, our lab thinks a lot about the so-called hidden risk, and we've always been fascinated by people like Churchill who have all of the wrong risk factors, smoking, obesity, stress, et cetera, but did not develop cardiovascular disease until well into his 90s. And what makes him different from people like Jim Fixx, who wrote the definitive exercise book many decades ago, was a fitness enthusiast, but as you know, was found dead on the side of the road while out running in his 50s. And so what is it that makes these two people different in their ability to to really go against their ingrained risk factors. And so this is where we've chosen to leverage unbiased human genetics. And we were very fortunate to get to work with pioneers like the late Jeanette Erdman, who was at the forefront of the GWAS, technology platform. And we now have dozens or maybe even hundreds of new genetically validated targets, many of which we think are causal for atherosclerosis. And the beautiful thing here is that many of these are independent of lipid metabolism. And so we have a lot of work to do to explain how they may cause disease. But the idea is that we now have new bona fide targets that can be investigated that we think may be responsible for this residual risk that occurs outside of the traditional risk factors like smoking or diabetes. Now, when I first started my laboratory, we were coming through the first wave of GWAS studies. And, what's fascinating is that nearly 2 decades later, the original findings still hold true. And the top GWAS hit for coronary disease resides at chromosome 9p21. You see this really remarkable hotspot of genetic risk here at chromosome 9 that rises well above all of the other signals that have been found. And, we liked this locus because not only are these very common risk alleles, so this isn't a rare disorder that affects, just a handful of patients, but actually 1 in 5 people in the US carries 2 copies of this risk allele. So the public health implications are profound. And also we know that this locus is completely independent of risk pathways. So this is not a gene that makes you more or less likely to smoke or be overweight or to exercise. This is something new that we thought if we could explain could really move the needle and help us develop new therapies. So we made knockout mice for the genes at this locus. It was really no surprise that the mice deficient in this gene had larger atherosclerotic plaques. This was predicted by the human genetics, and again was not a surprise. But what did stand out to us was the characteristics of the plaque. And in contrast to the control mice, which had very typical, very dense hypercellular lesions, we found the knockout mice had lesions that were almost completely replaced with what's known as the necrotic core. And so this is basically the graveyard within the plaque where apoptotic or inflamed cells will accumulate. And so we began to wonder if the reason that this 9p21 locus was promoting risk independently of established risk factors is because it might be regulating a process known as efferocytosis, which is from the Latin meaning to carry the dead to the grave. Now, this is basically just a fancy word for the phagocytic removal of inflamed or dying cells. And it turns out that this process is very densely regulated, and there are so-called find-me ligands that are basically chemoattractants that will be released from a dying cell to attract the macrophage to the site of inflammation. These cells will rapidly upregulate what are called eat-me ligands that will interact with receptors in the macrophage to help them interact. And then when this goes well, the corpse is rapidly ingested and degraded. And I think the key thing that happens after all of this is the upregulation of anti-inflammatory cytokines that we imagine is the way that the immune system says the danger signal has been controlled and that no additional inflammation is necessary. And what's now clear is that efferocytosis is not only involved in the removal of these cells, but also this late resolution of inflammation that we think is important for tissue healing and homeostasis. So in the ensuing years, we worked to map the signaling cascade here, and this is all published, but the takeaway is that we think that normally, when a cell undergoes programmed cell death, it will turn on these eat-me molecules, such as calreticulin, which will allow it to be recognized and removed. But we think that in people that have atherosclerosis or have these genetic variants, like those at 9p21, that they have a reduction in the expression of calreticulin, and that basically renders them invisible to the immune system. So they aren't recognized, they aren't removed. And then the real concern is that these cells will then undergo secondary necrosis, where all of this inflammatory debris can be released into the growing atherosclerotic plaque. And so now this concept is working its way into the textbooks, and the thought is that there's a defect in efferocytosis. This allows the apoptotic and necrotic cells to accumulate and the lesion to grow over time. But perhaps more concerningly, you have the secondary inflammation that will occur, and we think that this will destabilize the plaque and then promote the clinical events like heart attack and stroke. So this raises the question of how to reactivate this phenomenon. Well, we spent a lot of time trying to upregulate these eat me molecules like calreticulin. We ultimately came to the conclusion that this was going to be really hard to do on a cell that's already inflamed and undergoing cell death, and we might be better off going after the counterbalancing don't eat me signal, which in this case is a dominant negative signal called CD47. Our first question was whether these don't eat me molecules are present in the plaque in the first place. We did studies on rapid autopsy specimens, and we found that in healthy coronary arteries, these don't eat me molecules are expressed at a relatively low level. But in patients who had died of a heart attack, we saw really remarkable upregulation in the plaque, and particularly here in the necrotic core, shown with this red staining. We went on to confirm these protein findings at the RNA level with our colleagues at the Karolinska Institute, showing that these are increased in the carotid plaques of patients, both in their discovery and validation cohorts. And then we went on to show that this upregulation again occurs in reaction to inflammation, so that the TNF that's present in the plaque in the inflamed individual is driving the upregulation of the CD47. And this is the don't eat me signal that basically renders these cells invisible and prevents them from being removed. So our next question was whether we could target this process in vivo. And here we were able to take a page out of the oncology literature because it turns out that the very same don't eat me signals are turned on by cancers. And this is part of how they escape immune surveillance. Now, the oncologists had already hypothesized that interfering with this signal may be sufficient to reactivate the immune system and induce the removal of these unwanted cancer cells. In fact, our colleagues here at Stanford, led by Irv Weissman, had shown in a number of preclinical murine cancer models that blockade of this don't eat me signal could lead to a reduction or even a complete cure of cancer across a wide range of different tumor models. So we asked the very simple question of whether the same approach would work in atherosclerosis. And we treated mice with either a control antibody or one blocking CD47. Here you can see in the control animals that by EM that there are macrophages in the plaque which are seemingly ignoring all of this cellular debris shown off next to it with the yellow arrows. After we gave the blocking antibody, though, we could reactivate the appetite of these cells, and we found numerous examples where they would take up dozens of targets at the same time. Now, the removal of these unwanted and inflamed cells led to a significant reduction in the burden of plaque in these mice, shown here in the aortic sinus, and we found this throughout the entire body, including in the thoracic aorta. With the removal of these unwanted cells, we found that these mice were less inflamed, and the aortic signal, shown by PET-CT scan, was quenched in these animals. And not only were the plaques smaller and less inflamed, but they were less susceptible to plaque rupture. And so here we did a tandem stenosis model where we could induce intraplaque hemorrhage, shown here with this red blood cell stain in the plaque. And again, we found that after blockade of this signal, that we could reduce the plaque hemorrhage rate by about half. So this would lead to a reduction in clinical events in humans, according to our predictions. So now we have this cascade that we've mapped, again, which we think is related to inflammation, but we have shown that it's not a fixed defect, and that if we come in and block this axis, we can reactivate the immune system and induce regression of the plaque for the first time. So this then raised the question of whether we could translate these findings into humans. I mentioned that we were involved in the first oncology company that humanized these antibodies. And in their first clinical trial, they took patients who had been diagnosed with refractory lymphomas and had failed multiple lines of standard chemotherapies. and they gave them a blocking CD47 antibody as kind of a last line effort to try to treat their tumors. And what you can see here on the PET scans in this unblinded, non-placebo controlled trial, that there seemed to be a really impressive reduction in their tumor burden over time. But what stood out to us was that although these PET scans were done as a way to look for tumor growth and metastasis, that we could also quantify the inflammatory signal in the aorta, which you can see here in the aortic arch on the left-hand panel. And we had the hypothesis that while these patients' cancers were melting away, that perhaps they were also experiencing a reduction in their vascular inflammation. So to test this, we obtained these PET CT scans from these individuals. We de-identified them and anonymized them and had them reviewed by the head of nuclear medicine at Stanford. And what he showed us was that if you look at the signal in the carotid artery at baseline, which is pointed to by this arrow, you can see the signal here that's accumulating at the carotid bifurcation, which is where plaque will build up before patients have strokes. He found that after just 8 weeks of therapy, that this signal in the carotid was almost completely quenched. And regardless of how they quantified the inflammatory signal, whether they looked at the most inflamed segment or they looked throughout the entire carotid artery, that the signal was improved. And although this was retrospective and was not blinded, it was impressive that 8 out of the 9 patients seemed to show an improvement in their vascular inflammation, again, after just 2 months of therapy. So the question then is, why not stop there and just go straight into a clinical trial of actual cardiology patients? Well, one of the things that we worried about was whether we were going to have any off-target side effects from reactivation of the immune system, the same way that those anti-inflammatory therapies had immunosuppressive effects and the increase in inflammation and infections. Our first analysis was pretty reassuring. We found no non-specific toxicities with our therapies. The blood pressure in the mice was the same. The cholesterol was the same. Body weights and insulin resistance was the same. But we noticed that the spleens from these animals were significantly enlarged. And we went on to show that this was because there was a significant increase in what's known as erythrophagocytosis, where the macrophages in the spleen were eating the red blood cells and causing a clinically significant anemia. So while our therapy was very good at activating the immune systems and having the removal of the unwanted cells in the plaque, we also were unfortunately having the off-target clearance of healthy red blood cells in the spleen. And you can see here there was a significant reticulocytosis. The bone marrow is trying to rev up to keep up with this, but these animals were having a reduction in their blood count and an anemia, which we thought would be really, unacceptable to somebody with cardiovascular disease. Now, we had hoped that this was really a mouse-only phenomenon, but the group that did the first non-human primate studies observed the same pattern, and as they gave higher and higher doses of these therapies, the blood counts were dropping in these primates, including with several that were drifting down below the transfusion range. And indeed, in that first human clinical trial of cancer patients, they saw again, when they gave high-dose IV infusions of these medicines, that there was a drop in the hemoglobin. And again, this is probably okay if you have refractory cancer, you're willing to take a transfusion for this, but this is probably a toxicity that we can't accept for the garden variety atherosclerosis patient who's walking around with their subclinical cardiovascular disease. So we next turned our attention to trying to find a way to overcome this Achilles heel. And we thought about using nanotechnologies, particularly because of really pioneering work led by the late Sam Gambier here at Stanford, who ran the radiology department and had been developing nanotechnologies for the diagnosis of cancer and tumor metastasis. And his group had shown that a nanotherapy known as a single-walled carbon nanotube seemed to be specifically scavenged by the inflammatory monocytes and macrophages that were homing to the early tumor. And we had the idea that we could take these nanoparticles and use them as a Trojan horse, because we knew that they would get to the inflamed macrophage and potentially could be a way to deliver a payload of medicines exactly where we want them to be. So we adorn these nanoparticles with a number of chemical stabilizers and molecular imaging probes, and then pro-efferocytic therapies that were designed to enter the macrophage and then elute off and deliver the therapy directly to the site of inflammation and injury. We showed that in mouse models that just like our original antibody, these nanotherapies could potently reduce the atherosclerotic burden. And what was interesting was that in contrast to the antibody, which again would cause the anemia and the reticulocytosis, our nanotherapies had none of these toxicities. So we saw no reduction in hemoglobin, and we saw no increase in the reticulocyte count. And so this was a really, important first demonstration of the ability to use these Trojan horse nanoparticles to induce the effect in the diseased blood vessel without any off-target toxicity. And now, in collaboration with Lars Magnifessel in Germany, we've taken this into a large animal model. These are genetically modified pigs that develop atherosclerosis that's very similar to humans. And we were really happy to see that in this large animal model, our nanoparticles seem to have no toxicity. And although this model, really doesn't develop exactly the same type of plaque that humans do, we saw a reduction or a trend in reduction in the plaque burden in these early streaks in the coronary arteries of these animals. But most importantly, we saw that the inflammation that you can again see here in the carotid arteries of these animals seems to be quenched with our therapy. And so we've recently published these findings, and we're really excited because this technology has now been partnered with a company that's aiming to put them onto drug-eluting stents for the local delivery of these therapies at high concentrations directly to the atherosclerotic plaques in the coronaries. And so I'd say stay tuned for this. There have been some issues with nanotherapies, and so we have to make sure that the backbone of the Trojan horse itself is safe and non-toxic. But we're really excited and hope that this can become a new therapy in the future. But in the meantime, we've been thinking about how we could maybe build a better mousetrap, and if it's possible to make a second generation of antibody or antibody-like protein therapies that could be safer and more effective. Now, a lot of the stimulus for this came from the failure of first-generation therapies, where when they were advanced into larger phase 3 clinical trials for a variety of cancers, they found that the high doses of anti-CD47 therapy that were necessary to penetrate these tumors, led to, really intolerable hematological toxicities, including anemia and thrombocytopenia. So, our colleagues at Stanford have been working at building more potent CD47 blockers, including by taking the native receptor SIRP-alpha and making mutations in it to increase its binding efficiency. They were able to make a modified protein that had nearly a 50,000-fold increased binding affinity for CD47. And then we've now taken that molecule and grafted it onto a modified IgG4 backbone that has been adapted not only to increase its longevity, but also to inactivate the Fc-dependent functions. The idea here is that we now have a very potent CD47 blocker that's bound to a next generation antibody backbone that we think should not induce the Fc-dependent effects and not cause the hematological toxicities that were seen with the first generation antibodies. We've now advanced this into mouse studies, and we've seen, really beautiful reductions in plaque burden that occur in a dose-dependent manner. And earlier this year, we reported at ACC the results of the first in-human clinical trial, where we've given single doses in an SAD, single ascending dose manner. And we were really happy to see that at least single doses of this therapy were well tolerated, and we had no observed SAEs and really no significant anemia that rose to the level of causing concern. Based on the results from that study, we've recently initiated our first phase 2 study. This is the Matador trial, We're really excited about this. This is a repeat dosing, double-blind, placebo-controlled study where patients with atherosclerosis are going to be studied. And while this study is mainly looking at safety and pharmacology, we also are exploring whether or not this will have an effect on plaque burden and plaque inflammation. So we're really excited, and we hope that you'll stay tuned for the results of these studies, likely at the end of 2026. But in the meantime, while we're waiting for those results, we've tried to imagine what we're likely to find. And so to do this, we've been fortunate to partner with Gerard Pasterkamp and his colleagues, including Mikhail Mokri at the University Utrecht in the Netherlands. And we went to this group because Gerard oversees what I believe is the largest vascular biobank in Europe and perhaps in the world, known as AtherOXpress. And this is really a beautiful biobank with, I think, hundreds or maybe thousands of samples now taken from humans that have been very deeply characterized, both for pathology and clinical events, but also at a molecular level, where they've done multi-scale omics analyses and have shown that this can allow for molecular subphenotyping of plaques, both to assess the plaque composition, but also the plaque vulnerability and their association with clinical events. So we went to this group and we said, we, at least in a mouse model, knows what happens. We know what happens when we reactivate efferocytosis. And can we leverage the really rich genomic and phenotypic data in your biobank to try to model what might happen in humans that are treated with the same type of therapy? They were able to show that the signature of impaired efferocytosis does indeed correlate with one of the more inflamed plaque subtypes, and it correlates with the macrophage content in the lesion. They also found that the presence of an inflamed and impaired efferocytic signature correlates with future risk of symptoms, and those patients that were more likely to have a TIA or a stroke had evidence of impaired efferocytosis. And they've also generated a plaque vulnerability index score where they can try to predict the likelihood of plaque destabilization and rupture. And they found really a beautiful graded association between impaired efferocytosis and this plaque vulnerability index score overall. What was then really interesting was that using an AI algorithm, they were able to try to predict what would happen to the human plaques if they saw the same type of an effect that we've seen in our mouse models after the reactivation of efferocytosis. Now, I must admit, I don't exactly understand all that went into their informatic analysis, but what they found was that there is a prediction that the plaques would shift from a more vulnerable to a more stable phenotype after therapy, suggesting that not only would we reduce the size of the plaque, but we might stabilize the plaque, reducing the inflammation, thickening the fibrous cap, and making it less likely to rupture. Now, we wanted to make sure that this algorithm was producing predictable and plausible results. And so we then ran through a number of positive and negative controls through their same algorithm. First, we asked them to feed in a random list of genes, and we were reassured to see that they did not predict that alteration in that random list of genes would have any effect on plaque stabilization or destabilization. They then ran a positive control where they looked at gene profile data from mice that had been treated with endotoxin, which of course is a very potent inflammatory signal. And reassuringly, they found that this was predicted to destabilize the plaque and make it more likely to rupture. On the other hand, they came back and did another positive control with data from statin-treated animals. And as expected, they found that animals treated with lipid-lowering therapies and statins were predicted to have stabilization of their plaque. And then we were really pleased to see that the pro-efferocytic therapies seem to have at least as large, if not larger, beneficial impact as statins. And so, of course, we need to prospectively test and validate this. But these results will predict that our therapies may stabilize plaque at least as well as things like Lipitor. So As we wrap up, we're now, really excited because we think that this concept of of a defect in efferocytosis that in mouse models has been shown to promote not only the physical enlargement of the plaque, but also the inflammation in that plaque, likely is not only a reversible defect, but one that we predict will have benefit if we can be targeted safely in humans. We're excited because we think that we now have the ability to advance the next generation of anti-atherosclerotic therapies and to use human genetics to move beyond the traditional risk factors to finally be able to address this residual risk that we've not been able to really treat over the last several decades. We agree with those that think that targeting inflammation is going to be the next major therapeutic horizon for our field. And that's why we're so excited about efferocytosis, because not only does it improve the physical obstruction that's developing the plaque, but we think because phagocytosis is so closely intertwined with anti-inflammatory effects, that this will be able to directly suppress inflammation, but do it in a way that we don't make the animal susceptible to opportunistic infections, and hopefully will allow us to avoid increase in infections and septic complications in our clinical patients as well. We think that by blending disciplines and thinking across traditional silos, incorporating learnings from nanomedicine, oncology, and immunology, we will be able to move to this era of precision therapies for patients and hopefully do so with enhanced efficacy and reduced toxicity. So please stay tuned. We have a lot of fun things coming on the horizon in 2026. So with that, let me just acknowledge the group that did the work in the lab. We have a really remarkable group of trainees and postdocs who have helped advance these concepts over the years. I have to thank and acknowledge my fundings from the NIH, the American Heart Association, the Great House Foundation, and the Johnson family. John, thank you again for this really wonderful opportunity. I'd be happy to take any questions if we have time.
Thank you, Nick. That was really interesting, wasn't it? I love the way you took this all the way from basic science observation, from the GWAS studies and your basic science observations, all the way to translational models and to the clinical observations. Really wonderful work and a promising new therapy. So what you told us today was, you told us that these don't eat me signals in the plaque account for the growth of the plaque, its unstable state, its necrotic core, and by interfering with these don't eat me signals that you can potentially remodel the plaque, reduce its size, make it more stable, and reduce the major adverse cardiovascular events as a result. Really interesting. One of the things you talked about was an antibody against CD47. So CD47 is the receptor, I guess, that recognizes the don't eat me signal.
Yeah, so CD47 is the ligand that's expressed on the cells. The receptor is a molecule called SIRP alpha. CD47 is very highly conserved, and we think that it was, one of the ways that our body separates self from non-self. Our immune system has the tall task of removing billions and billions of cells every day that are turned over through normal homeostatic processes. Think about as our hair grows, think about as our gut is turned over, think about all the cells turning over in our bone marrow. That normal turnover has to be handled, and so the garbage has to be taken out. And so our immune system is really good at identifying and removing those aged, senescent, and unwanted cells. It also uses the same concepts to recognize non-self, like bacteria or other infectious agents. And it's one of the ways that our body recognizes pathological cells like tumors. But the body has to be very careful not to get rid of healthy cells, the self that needs to persist. And so CD47 is one of those signals out of many that is expressed, and it tells the immune system, hey, I'm part of the healthy body. I am not ready to be removed. And so the body is very careful about how it regulates these eat me and don't eat me signals to govern the removal of those cells. And we find that when CD47 becomes overexpressed or doesn't get turned off on cells that are dying and need to be recognized for removal. And so that's why we think that going after, that imbalance is going to be the way to hopefully to help cancers, and then also help people that have plaque building up through the same mechanism.
CD47, my understanding, is also expressed by senescent cells. So a problem with senescence is senescent cells make the other cells around them sick by secreting inflammatory cytokines and disturbing signaling. Could this also be a senolytic therapy targeting CD47?
I think it could, John. It's a great question. Our lab doesn't look at that directly, but I've heard tell that other people are looking into this, and so I think that it absolutely could have a role as a senolytic in the future. So I think stay tuned for some exciting data that may be coming out from other groups on that topic.
You made an antibody against CD47, and it looks promising. I wonder if you've thought about also making CAR T cells that are expressing the antibody against CD47. And the reason I ask you that is that there's some evidence that antibodies, for example, against, like rituximab, against pathogenic B cells, they're effective, but they don't clear all the pathogenic B cells out of the tissue, whereas CAR T cells directed against pathogenic B cells can clear pathogenic B cells out of the lymph nodes and are more effective. So is that another direction you might pursue?
Yeah, it's a great idea. Our lab doesn't work on T cell biology much, but I do know that many people have suggested that CD47 and efferocytic pathways may be involved in T cell biology. One thing that we've thought about is expanding the concept of CAR T to CAR M, and I know that there are groups that are trying to engineer macrophages to train them to be better killers and better phagocytes, and so I think that you may see some of that coming down the pike as well. Our lab hasn't gotten there yet, but I think it's a fantastic idea.
And one last question, and I know Anita has some questions for you as well. I think this is also an area that would be ripe for vaccines. So, we're making mRNA vaccines here at Houston Methodist. We have a Center for RNA Therapeutics and an active RNA core. We're actually making vaccines against a number of infectious diseases and cancer. We've got an IND for triple-negative breast cancer. We're starting the study next month. What about a vaccine against CD47?
No, it's a good idea. I hadn't thought about it. I mean, the caveat that I was trying to convey throughout the story is that this is a pretty delicate and very important biological mechanism, and so it really does seem to have a Goldilocks zone, and you have to get the dose and the titration just right between the eat me and don't eat me signals. And what we've seen so far is that anytime you step on the gas too much or you take away the brake too much, that while you might get the benefit you're looking for, this can come with some toxicity and removal of the healthy cells. Yeah. So that's why we're spending so much time thinking about trying to deliver it just to the inflamed area, or do it in a way where we're not activating the unwanted aspects of the immune system. I just don't know enough about vaccine biology to know if you can titrate the effect the way you might need to. But again, if that's doable, if you could tune that the way you wanted, I think that it could have a lot of potential.
Thanks. Go ahead.
Sure, that was a really fascinating talk. I mean, a couple of questions about the senescent Dr. Cook already asked, but I was also wondering if these CD47 overexpression cells, do they express P21, P16? Obviously, you mentioned they are secreting more inflammatory cytokines, but anything about like a beta-gal activity or?
Yeah, these are great questions. We haven't looked specifically, and we haven't used the aged mouse models, which I think that we should. The more I'm listening to this discussion, the more I think I need to go back and start thinking more about this possibility. We haven't looked, but the link between senolysis and senolytics makes a lot of sense, and it's certainly an area for future research.
Other question, could you elaborate on that variant of chromosome 9? You mentioned that they are more prone for atherosclerosis. Is it that variation associated with other hallmark of aging, or is it so specific to atherosclerosis, or is it more prone for any vascular disease?
Yeah, that's a great question. 9P21 is really a fascinating a hit that is still to this day not well understood, which is shocking when you think about it being by far the most significant association that's ever been associated with coronary disease. It turns out that it's a marker of pan-vascular risk, so having this puts you at risk for coronary disease, PAD, stroke as well. And what's fascinating is that it's also linked to non-atherosclerotic forms of vascular disease, including aortic aneurysms and even intracranial aneurysms. And that's important because AAA, of course, overlaps with atherosclerosis in many patients. And so you wonder if it's really just athero, that's the signal being found. But the fact that you're also seeing the link to structural aneurysms in the brain makes us really believe that this is not just a lipid pathway or something related to the usual biology, but something structural. And so we think that this may have to do with smooth muscle physiology. Our group has studied this quite extensively, and it's linked to perhaps enhanced apoptosis in the vessel wall. And so we think that that may be inducing the inflammation that's driving the atherosclerosis and also promoting the medial thinning that can be observed in aneurysms. I don't know about links to other forms of vascular disease, but 9P21 is absolutely an area that I think has not been fully explained and still is an area where more biology could be learned and more therapies could be developed.
Great. Maybe just one more question. You showed the data that if you use antibody consistent for a long time, the mice at least showing the larger spleen and the toxicity associated with that. I wonder, I'm just saying that based on what I have learned with the senolytic drug, they do it as a hit and run, so you don't use it all the time. And I would imagine that what you see in the spleen can be recovered. So if you use it, do you have to use it all the time consistently, or can you use it the way that senolytic drug can? They're using that in the clinic.
Yeah, it's a great question. I mean, I think that for the oncologists, they're so focused on getting rid of as much cancer as possible, but atherosclerosis is a disease, as you know, that incubates for decades. We start developing plaque in our 20s, and then it may not manifest for years or decades to come. I think that there could be a role for intermittent dosing. I think there could be a role for single dosing at the time of a myocardial infarction when inflammation is most pronounced and you wanted to cool off the vessel. I didn't mention it here, but there's also very exciting data that it may help remove dying cardiomyocytes. And a number of groups have shown that this can be a useful approach for ischemic cardiomyopathy and helping with reperfusion injury. So there is a real scenario that we've been thinking about where it could be given as a way to prevent post-infarct heart failure. So maybe this could become a single dose given at the time of acute MI in the cath lab to prevent that remodeling that you don't want, that scarring that you don't want. So I think that there are ways to be creative, either with intermittent dosing or with some of these targeted nanoparticles. So, again, stay tuned. We have a lot of ideas coming down the pike.
Great,
Nick, thanks so much. We've reached the end of our program. We've been hearing from Dr. Nick Leeper today from Stanford University talking about his basic insights into efferocytosis, atherosclerosis, and the promising avenue for therapy in the future. Nick, thank you for joining us on the show.
Thank you.
Thank you for having me.